Okay, seniors, this is the second part of the screencast review that was requested. This is requested by Armor, the motion review. He wants to focus on muscle contraction, but I'm not sure, so I'm just going to go through and do both parts, all parts of 11.2. Okay, so first things first, we're going to talk about bones and exoskeletons, how they act as connection points for muscles that act as levers. Different kinds of joints. We'll talk about pairing of muscles, antagonistic pairs that we talked about in the respiratory system as well. Talk about skeletal muscle cells, how they're different from others. It connects us back to topic 1. What the structure of muscle fibers, and then applications will be antagonistic muscle pairs and human to elbow annotation. So this musculoskeletal system is an important system. We often don't think about it. The purpose, of course, is to provide support, protection, and attachment points for muscle. You need to have attachment points for muscles to pull on so that you can move. No bones, no movement. Okay. We also need to compare endoskeletons and exoskeletons. So of course, endo means inside and exo means outside. So we have an endoskeleton, it's inside of our body, where a lot of insects, crustaceans, those kind of things, they have exoskeletons. It is outside of their body. But both of them work in pretty much the same way. They just are different attachment points, either inside or outside. So the example we'll look at here was the grasshopper, and it can jump really high compared to its body size, which is really impressive, and it's because of the way its leg joints are put together. So the tibia muscle, it's a big muscle here in its upper leg, right here. It contains two muscle pairs that work together, the tibia extensor and the tibia flexor. And how they work together is they both act on the same bone on the tibia. So if you look here, here's the flexor and here's the extensor, and they contract to move the leg and allow it to jump really high. The muscle strength and bone strength compared to the size of the insect is very impressive. Other pairs we have, of course, the classic one everyone thinks about is bicep and tricep. As one is contracts, the other relaxes. As the other one contracts, the other relaxes. This is how muscles work, because muscles have to be paired up because they cannot pull. So they cannot push, they can only pull. Muscles can only move in one direction. So to move your arm up and then move it down, you have to have a muscle for each motion. Biceps move your arm up and triceps move your arm down. Different muscles to do that. So here's a whole list of pairs. As long as you name a couple pairs examples, you're probably good to go. You are good to go, sorry. Okay, key ideas. The musculoskeletal system provides protection, support, and an anchor point for antagonistic muscle pairs. Muscles can only pull in one direction, so they must work in pairs to achieve opposite motions. These systems work similarly in organisms with an exoskeleton as does with an endoskeleton. Okay, so remember about zygote body, you can go there and look at all the different systems. A really useful review tool for topic 6 and topic 11, so feel free to use that. And then look at different questions, the different joints, ligaments, that kind of stuff. So the types of joints you need to know for this course are synovial joints, and synovial joints are bone-to-bone joints that contain synovial fluid, and that's between the bones to help lubricate, help keep everything from being worn down, keep it safe. It's also used for cushioning, and a lot of the time the capsa bones are covered in cartilage and helps also with cushioning, just like the synovial fluid does. There are 6 different types of synovial joints. Here they are here in this image. Saddle joints, plane joints, hinge, ball and socket, ellipsoid, and pivot joints. So take a look and think about how the different kinds of joints allow different kinds of movement. Here's the elbow joint, which we're talking about, and that is an example of a hinge joint. So here's the elbow, and again, you have to be able to annotate this diagram. So that means they'll give you a diagram, and you'd have to label it. You're not really going to have to do that on the RAS file. It's not really a thinking kind of question, but it might ask you to apply the information about the elbow to talk about something else. Okay. So there's little answers. Key ideas. The structure of synovial joints are designed to reduce impacts between bones while still allowing for strong connections between bone and muscles. Ligaments connect bones to other bones at joints, while tendons connect muscles to bones. Okay, different kinds of muscle types. Connecting back to topic 1. So we have the skeletal muscle, cardiac muscle, and smooth muscle. So they have different types, and if you notice, in topic 1 we talked about skeletal muscle here, having all these nuclei, because a lot of them is skeletal muscle, and it's different than the other 2 kinds of muscle cells. Okay. So skeletal muscles are one of the exceptions because they have so many nuclei, and they have to have so many nuclei because they need to make lots of protein. And the instructions for making protein are in the DNA, which is in the nucleus. So more instructions means more transcription. Translation means more proteins can be produced. They also have what's called a sarcoplasmic reticulum, a specialized type of endoplasmic reticulum that's really important for the release of calcium during muscle contractions, which we're going to talk about in a little bit. Okay, so skeletal muscle has many nuclei and sarcoplasmic reticulum to provide them with sufficient proteins and energy to perform their task of muscle contraction. Okay, so let's talk about muscle contraction. So muscle fibers contain many myofibrils. They're made of contractile sarcomeres. And the contraction is achieved by sliding of actin and myosin filaments. ATP hydrolysis and cross-bridge formation are necessary for the filaments to slide. We use calcium ions and the proteins tropomyosin and troponin to control muscle contractions. You have to be able to draw and label a sarcomere and analyze electron micrographs to find the state of contraction of muscle fibers. That's more likely question to show up, the analysis type of question, than a drawing question for your RAS final. Okay, so here's the diagram. You saw this one earlier of a skeletal muscle. So we have here the muscle fiber, many of them coming together to make a fascio, many of those coming together to make these bigger group punches here, and together makes a big piece of muscle, which is attached to a bone with a tendon. Okay, the idea is that these muscle fibers here are made of myofibrils. So if we zoom in even further, here's that single muscle fiber, which in the last diagram was this tiny little piece. Now we're zooming in on that. Okay, so that's this. And that muscle fiber is made of many of these myofibrils. And a myofibril, if you zoom in on that, looks like this. So we have these sarcomeres, which kind of goes from Z line to Z line. Here, that's a sarcomere. And that's where the contraction is going to be happening. Okay. So to draw a sarcomere, make sure you look, and they're good to make sure you analyze as well. You don't have to draw for the RAS final because that's not a thinking skill, more just a memorization skill. And you'll notice how when they are relaxed, they're much longer. And when they're contracted, it's kind of this gap on the ends here in the middle. And when they're contracted, it's a much smaller gap. There's no gap here. Everything's kind of closer to each other. Okay. So skeletal muscle is fibrous, and each fiber is made of myofibrils, which are then made of contractile sarcomeres. The structure allows for the microscopic contractions of sarcomeres to combine into whole muscle contraction. So when you're moving your body, it's happening on a scale of millions of sarcomeres contracting at the same time, which, if you think about it, is really cool. Okay, so let's talk about myosin and actin. So this happens because myosin is acting on actin. And if you look at the structure here, you'll notice the myosin has these distinctive head-like structures, right? So these little round kind of circles, that's the myosin heads. And the thin fibers, the blue fibers here, the thin ones, those are actin. So the myosin heads grab onto the actin. Okay, and then if you look here, the sarcomere is longer when it's relaxed and shorter when it's contracted. That's because it's sliding, the myosin is sliding along the actin. It's pulling the actin along. And the theory we use to explain how this works is called the sliding filament theory of muscle contraction. Okay, so we have myosin here. Here's our myosin heads acting on actin, which is the yellow fibers here. Okay, so myosin is one continuous protein. It cannot get shorter. If you look at the whole sarcomere diagram, like here, this myosin is one entire thing, right? It's not broken up. There's no gaps in it. When like there's gaps here there's gaps in the actin, but there's none in the myosin, so it cannot really get shorter. So what happens is to connect the Z lines, the actin is connected by the Z lines on both ends, and then you can pull on the actin and pull the entire thing to each other. And you'll tell muscles contracting when the two Z lines on both ends of the sarcomere are being pulled closer, and that's a contraction of the muscle. This happens when myosin heads are activated. And the myosin heads here, they can be activated when they have one ATP is converted to ADP and a phosphate. And that allows them to reach out to try and grab the actin. And that connection between myosin and actin is called a cross bridge. And when they're done with that, they release the phosphate group. Okay, and once the ADP is then going to be released after it's now bound to the actin, and that energy in the ATPs will be used by releasing the ADP, that releases energy, which allows the myosin to move forward and pull the actin towards the center of the sarcomere. And then the myosin will bind to a new ATP and be released from the actin. It can go back down again for another contraction or just to wait until the signal comes from the nervous system for the next contraction. Okay, so myosin heads bind to the active site of actin filaments, creating a cross bridge, and then pull it towards the M line at the center of the sarcomere. So it's a good idea to have a sarcomere image in your mind when you're thinking of this. The movement is controlled by the hydrolysis of ATP to ADP, the release of the phosphate group, and then the release of ADP. Okay, so how is this controlled? What's the signal happening here? A motor neuron, a neuron that connects to skeletal muscle, that is the signal that it needs. So acetylcholine is our main neurotransmitter you need to know for this course. When it's released in a motor neuron, it's going to go to the sarcolemma and bind down here. Okay, this is going to open the sodium channels. And the sodium channels are going to change the voltage. It opens these sodium channels, and that was going to cause a motion through the T tubules here and releases calcium. So calcium, remember, is stored in the sarcoplasmic reticulum. It's going to be released and then given out here into the sarcomere and allows it to bind, the myosin to bind to the actin. So here we go. Here we have the T tubules here on both ends of the sarcomere. The motor neuron is going to be releasing this neurotransmitter, acetylcholine, which causes calcium to be released, go down here, then released from this part here, from the sarcoplasmic reticulum into the myosin, into the sarcomere. Okay, and then it can contract. Move the two Z lines, one Z line here, one Z line here, closer to the M line in the middle. Okay, so we need to have calcium to send that signal. And then, so when that's happening, the myosin heads, they're bound with ATP, they're ready. Remember, they can't bind to actin until they turns ATP into ADP. And they wait the signal from the next neuron. So here's a little animation, so you can see that happening. The signal's coming down the neuron. It's going to release the neurotransmitter. That's then going to send the signal down the T tubule, which releases calcium into the sarcomere, which binds and allows the contraction to happen. Okay, it's a great animation from BioNinja. Take a look at their stuff on there too to help you if you want. Okay, so why is calcium necessary? What does calcium actually do? Well, what happens, if you look in this diagram here, what I was showing you earlier, this myosin head can't actually attach to actin. Actin is this thin yellow dot kind of fiber here, and there's this green thing blocking the way. Myosin can't actually attach to actin because troponin, these green fibers, which are attached to the fiber of tropomyosin, it's blocking it. It can't physically attach. So what happens is the troponin, these green ones, they have binding sites for calcium. So when calcium is released, it's going to bind with troponin and cause the whole thing to change its shape. And when it changes its shape, it's going to uncover the actin, the binding sites on actin. So myosin then bind to actin. So here's another kind of image that here's a myosin trying to bind to actin, but it can't because tropomyosin is in the way and troponin as well. When calcium is released from a signal, from a sarcoplasmic reticulum, from a neuron signal, then it's going to bind to troponin and shift the entire structure, kind of slide it away, so that myosin can now bind directly to actin and slide. Okay, so when acetylcholine enters the synapse of a neuromuscular junction, it triggers the sodium channels in the muscle, which in turn causes calcium to enter the sarcoplasm. The calcium binds the troponin fibers, changing the shape of tropomyosin and exposing the active sites of actin, thereby allowing myosin to bind and then muscle to contract. You need to be familiar with all these terms. On the exam, you're going to have so much to get through so quickly, you can't be looking up what these words mean. You need to know them. Okay, so there's a little video showing how it works. I'm not going to play that for you now. You can look it up if you want to. And then we went through and did a practice activity with this, where here's kind of the steps, where we're going to have calcium ions released by a sarcoplasmic reticulum. They cause cross bridges to form between actin and myosin by releasing the tropomyosin block. The energy in the head of the myosin moves the head, and that slides the actin past the myosin, shortening the muscle. That's the contraction as the muscle gets shorter. The ATP is used to disconnect myosin head from the actin, and it kind of resets it for the next contraction on the next signal. So we went through these step by step by step in a group. You should also look at electromicrographs. This is a more likely kind of question I'm going to give you, is to look at some diagrams to analyze. So here we have a relaxed sarcomere, and here we have a contracted sarcomere. You can tell by the distance from the Z lines over here to the M line in the middle. This is much closer, much further apart. Okay, and again, we have a relaxed and contracted muscle fiber. Okay, key ideas. When examining skeletal muscle under the microscope, and its contracted or relaxed status can be determined by examining the distance between the Z lines. In a relaxed muscle, they are much further apart than in a contracted muscle. And that is all you need to know for 11.2, muscle contraction. Hope this is helpful for you. If you have any questions, message me. I'm happy to do a one-on-one Zoom, check in with you, see if you have any questions, and help you prepare for your RAS final.